Medical teaching script. Biochemical events in the nucleus and nucleotide metabolism. A narration-ready teaching script based on basic biomedical sciences. Block 2, Module 2, Case 1. COVID-19, Quo Vadis. The big picture. The title of this module refers to COVID-19, but the pages provided are mainly about a more foundational subject. They explain how genetic information is stored in deoxyribonucleic acid, or DNA, how that information is copied into ribonucleic acid, or RNA, how RNA guides protein synthesis, and how cells manufacture, recycle, and degrade the purine and pyrimidine nucleotides needed for these processes. This foundation matters in every area of medicine, including virology, because viruses and human cells both depend on nucleic acids and nucleotide chemistry. However, because the supplied notes do not directly describe the life cycle of SARS coronavirus 2, this script will not add a separate viral life cycle discussion that is absent from the source. Think of the topic as one continuous journey. We will begin inside the nucleus with the structure of DNA. We will then follow information from DNA to RNA during transcription, and from messenger RNA to protein during translation. After that, we will examine how DNA is duplicated before a cell divides. We will then study the different types of RNA, the digestion of dietary nucleic acids, and the synthesis, salvage, regulation, and degradation of purine and pyrimidine nucleotides. Finally, we will return to the basic language of bases, nucleosides, nucleotides, and nucleic acids, ending with Chargaff's findings about DNA base composition. Biochemical events in the nucleus. Inside the nucleus, genes are arranged one after another along extremely long DNA molecules. DNA means deoxyribonucleic acid. A human DNA molecule can be enormously large, with a molecular mass measured in the billions of Daltons. Despite that size, it is built from a small collection of simple chemical components joined in a regular pattern. The extraordinary complexity of genetic information, therefore, comes mainly from the order of these components, not from a huge variety of different building blocks. The first building block to understand is the nucleotide. Each DNA nucleotide contains 3 components. It contains a phosphate group, the 5-carbon sugar deoxyribose, and 1 nitrogen-containing base. The 4 bases used in DNA are adenine, guanine, cytosine, and thymine. Adenine and guanine are purines, which have 2 fused rings. Cytosine and thymine are pyrimidines, which have 1 ring. The supplied notes use the older acid names deoxyadenilic acid, deoxythymidilic acid, deoxyguanylic acid, and deoxycitidilic acid. In modern biochemical language, these are more commonly described as deoxyadenosine monophosphate, deoxythymidine monophosphate, deoxyguanosine monophosphate, and deoxycitidine monophosphate. They may be shortened to DMP, DTMP, DGMP, and DCMP, but the full names are clearer for narration. Many nucleotides join to make a D and a strand. The phosphate of one nucleotide connects to the sugar of the next, producing a repeating sugar-phosphate backbone. The bases project inward from this backbone. A second D and a strand lies opposite the first, and the bases on the 2 strands form specific pairs. Adenine pairs with thymine, while guanine pairs with cytosine. Adenine and thymine are held together by 2 hydrogen bonds. Guanine and cytosine are held together by 3 hydrogen bonds. These hydrogen bonds are individually weak enough to be separated when DNA must be copied or read. But the large number of bonds across a DNA molecule gives the complete double helix considerable stability. The 2 strands are complementary. If one strand contains cytosine followed by guanine, the opposite strand must contain guanine followed by cytosine. If one strand contains thymine followed by adenine, the opposite strand must contain adenine followed by thymine. This complementary relationship is what allows one strand to serve as a template for constructing the other. The 2 complementary strands twist around each other to form the double helix. The notes describe 10 base pairs in each full turn. A more precise value for the common B form of DN is approximately 10.5 base pairs per turn, although 10 is a useful rounded teaching value. The important idea is that the double helix is not a random coil. It has a regular geometry created by the sugar phosphate backbones on the outside and the paired bases on the inside. The genetic code. The medical importance of DN lies in its ability to control which proteins a cell produces. It does this through the genetic code. The order of bases in DN stores information. When a gene is expressed, the two DN strands separate over that region, exposing the bases so that one strand can serve as a template. Genetic information is read in groups of 3 bases. Each 3-base unit is called a triplet when we are referring to DNA, and a codon when we are referring to messenger RNA. A codon specifies an amino acid or provides a start or stop instruction. A succession of codons therefore determines the order of amino acids in a protein. The example in the notes begins with the DN template triplets GGC, a GA, and CTT. Because messenger RN is complementary to the template, these produce the messenger RN codon CCG, UCU, and GA. CCG codes for proline, UCU codes for serine, and GA codes for glutamic acid, also called glutamate. This example works only if the displayed DN sequence is understood to be the template strand. That template versus coding strand distinction is a common examination trap. Transcription, moving information from DN into RNA. DN remains mainly inside the nucleus, but many cellular activities occur in the cytoplasm. The cell therefore needs an intermediary that can carry genetic instructions from nuclear DN to the cytoplasmic machinery that makes proteins. That intermediary is RNA. The process of making an RN a copy from a DN a template is called transcription. During transcription, a limited region of the DN, a double helix, opens. One DN, a strand, serves as the template. RN, nucleotides are joined in an order complementary to that template. If the DN, a template, contains guanine, the RN receives cytosine. If the template contains cytosine, the RN receives guanine. If the template contains adenine, the RN receives uracil. If the template contains thymine, the RN receives adenine. RN resembles DN but has 2 major chemical differences. First, RNA contains ribose instead of deoxyribose. Ribose has a hydroxyl group on its 2' carbon that deoxyribose lacks. The notes call this an extra hydroxyl ion, but hydroxyl group is the medically accurate term. Second, RN uses the pyrimidine base uracil in place of thymine. Its 4 main bases are therefore adenine, guanine, cytosine, and uracil. The immediate substrates for RNA synthesis are ribonucleoside triphosphates. These are adenosine triphosphate, guanosine triphosphate, cytidine triphosphate, and uridine triphosphate. They may be spoken as a TP, GTP, CTP, and UTP. Each incoming nucleotide carries 3 phosphates. During polymerization, the terminal 2 phosphates leave together as pyrophosphate, and the remaining nucleotide becomes covalently incorporated into the growing RNA chain. Subsequent breakdown of pyrophosphate helps drive the reaction forward. The notes describe this as activation of RN nucleotides by RN polymerase. A useful correction is that the nucleoside triphosphates are already energy-rich substrates when they reach the polymerase. RN polymerase selects the correct nucleotide, catalyzes formation of the phosphodiester bond, and releases pyrophosphate. It does not first create the triphosphate form by adding phosphates to each nucleotide at the gene. Transcription begins when RN polymerase and associated transcription factors recognize a promoter. A promoter is a DN sequence located near the beginning of a gene. In eukaryotic cells, transcription factors help recruit the appropriate RN polymerase to this promoter. Once positioned, the enzyme opens a short region of DN, and moves along the template strand. As RNA polymerase advances, it reads the DNA template in the 3' to 5' direction and synthesizes RNA in the 5' to 3' direction. The phrase 5' refers to carbon number 5 of the sugar, while 3' refers to carbon number 3. At each step, a complementary RNA nucleotide briefly pairs with the exposed DNA base. The polymerase then joins that nucleotide to the 3' end of the growing RNA chain. Only a short DNA region remains open at one time. Behind the polymerase, the newly made RNA separates from the template, and the two DNA strands reform their original double helix. When the polymerase reaches termination signals, transcription ends and the RNA transcript is released. The simple chain-terminating sequence description in the notes is especially accurate for bacteria. Termination in human cells is more complex and depends on the type of RNA polymerase and on RNA processing signals. The result is that the information stored in DNA has been transferred into a complementary RNA sequence. For a protein-coding gene in a human cell, the first product is usually precursor messenger RNA. This precursor is processed in the nucleus before mature messenger RNA passes through a nuclear pore into the cytoplasm. Translation, turning messenger RNA into protein. Translation is the process by which ribosomes read messenger RNA and assemble amino acids into a protein. Messenger RNA is read from its 5' end toward its 3' end. Translation normally begins at a start codon, usually a UG. This codon establishes the reading frame and usually introduces methionine as the first amino acid. As the messenger RNA passes through the ribosome, each codon is matched by a transfer RNA molecule. Transfer RNA contains a 3-base anticodon that is complementary to a messenger RNA codon. The transfer RNA also carries the amino acid specified by that codon. In this way, the nucleotide language of messenger RNA is converted into the amino acid language of protein. Translation continues until a stop codon enters the ribosome. The 3 stop codons are UAA, UAG, and UGA. They do not recruit an amino acid carrying transfer RNA. Instead, they recruit release factors that promote release of the completed polypeptide. A single messenger RNA can be translated by several ribosomes at the same time. The notes describe clusters of approximately 3 to 10 ribosomes on one messenger RNA, although the number can vary and can be larger. Such a cluster is called a polyribosome or polysome. Each ribosome is at a different point along the same messenger RNA, so many copies of the same protein can be produced simultaneously. Ribosomes are not specific for one particular protein. A ribosome can translate any messenger RNA that presents the proper signals. Specificity comes from the sequence of the messenger RNA, the transfer RNA molecules, and the genetic code, not from having a separate ribosome for every protein. Some ribosomes remain free in the cytosol, while others become associated with the rough endoplasmic reticulum. The destination is determined by the growing protein. If the new polypeptide begins with an endoplasmic reticulum signal sequence, a signal recognition particle binds that sequence and directs the ribosome to a receptor on the endoplasmic reticulum membrane. Translation then continues with the growing protein, passing into the endoplasmic reticulum lumen or becoming inserted into its membrane. Proteins entering this pathway commonly include secreted proteins, lysosomal proteins, and many membrane proteins. Proteins without the appropriate signal sequence are usually synthesized on free ribosomes and remain in the cytosol or are later directed to another destination. Therefore, the distinction is not simply glandular cells versus non-glandular cells. The decisive factor is the targeting information in the protein itself. Before an amino acid can be used in translation, it must be attached to the correct transfer RNA. An aminosyl transfer RNA synthetase first reacts with the amino acid and adenosine triphosphate, ATP is converted to adenosine monophosphate, or a MP, plus pyrophosphate. Because pyrophosphate is then split, this activation uses the equivalent of two high-energy phosphate bonds. The activated amino acid is transferred to its specific transfer RNA, producing an aminosyl transfer RNA. The charge transfer RNA enters the ribosome. Its anticodon pairs with the appropriate messenger RNA codon, placing its amino acid in the correct position. The ribosome then forms a peptide bond between the growing chain and the newly delivered amino acid. The notes describe peptidyl transferase as a ribosomal protein. The central correction is that the catalytic peptidyl transferase center is formed mainly by ribosomal RNA in the large ribosomal subunit. In other words, the ribosome acts as a ribozyme. The protein components help construct and stabilize the ribosome, but ribosomal RNA performs the key catalytic chemistry. The energy cost is high. Charging one transfer RNA consumes the equivalent of two high-energy phosphate bonds because ATP becomes a MP. During elongation, guanosine triphosphate, or GTP, is also used for accurate transfer RNA delivery and for ribosomal translocation. The usual high-yield total is therefore about 4 high-energy phosphate bonds for each amino acid added to a growing protein. This is why protein synthesis is one of the cell's most energy-demanding processes. A peptide bond joins the carboxyl group of one amino acid to the amino group of the next. The net chemical result is equivalent to removing the elements of water, meaning one hydroxyl component and one hydrogen component, while joining the two amino acids. It is better not to imagine that a free hydroxide ion and a free hydrogen ion are simply pulled out in isolation. In the ribosome, the reaction is a precisely catalyzed transfer involving the energy-rich bond that attaches the growing peptide to transfer RNA. DNA in cell reproduction. DNA does more than direct protein synthesis. Genes and their regulatory sequences also influence cell growth, cell specialization, and whether a cell proceeds through division. From the fertilized ovum to the adult body, controlled use of genetic information guides development and tissue maintenance. The cell cycle is the interval from one cell division to the next. In rapidly proliferating mammalian cells, the complete cycle may take roughly 10 to 30 hours, although the timing varies greatly by cell type and physiologic condition. Mitosis, the stage in which duplicated chromosomes are separated, may occupy only about 30 minutes in some rapidly dividing cells. Most of the cycle is therefore interphase. Interphase includes the G1 phase, when the cell grows and prepares for DNA synthesis, the S phase, when DNA is replicated, and the G2 phase, when the cell prepares for mitosis. Highly stimulated bone marrow cells can cycle quickly, while many mature neurons remain outside the active cycle for the lifetime of the person. The note's central lesson is that inhibitory and stimulatory signals strongly influence whether cells proceed through the cycle. Before mitosis can occur, every chromosome must be duplicated. The notes state that DNA synthesis begins approximately 5 to 10 hours before mitosis and lasts about 4 to 8 hours, followed by another 1 to 2 hours before mitosis. These values are approximate examples, not fixed times for every human cell. Their purpose is to show that replication occupies a major preparatory phase and must be completed before chromosome segregation. DNA replication. DNA replication follows the same basic complementary base principle used in transcription, but the goal is different. Transcription copies a selected gene into RNA, usually using only one DNA strand as the template for a particular transcript. Replication copies the entire DNA molecule, and both parental strands serve as templates. Replication begins at origins and proceeds through Y-shaped regions called replication forks. Helicase separates the two parental strands by disrupting the hydrogen bonds between complementary bases. Single-strand binding proteins stabilize the exposed strands. Topoisomerases act ahead of the fork. They temporarily break and then reseal one or both DNA backbones, relieving torsional strain and preventing excessive overwinding. DNA strands are antiparallel. One runs from 5' to 3', while its partner runs from 3' to 5'. DNA polymerases can add nucleotides only to a free 3' hydroxyl group, so every new DNA strand is synthesized in the 5' to 3' direction. This creates the leading strand and lagging strand problem. On the template that runs 3' to 5' toward the fork, the new leading strand can be synthesized continuously in the same overall direction as fork movement. On the opposite template, the new lagging strand must be synthesized discontinuously, away from the fork in short stretches called Okazaki fragments. DNA polymerase cannot begin a new strand by itself. Primase first makes a short RNA primer that provides the required free 3' hydroxyl group. The leading strand generally needs one primer at an origin for each replication unit, while the lagging strand needs a new primer for every Okazaki fragment. The notes say that lagging strand primers are only several bases apart. That is too short. In eukaryotic cells, Okazaki fragments are commonly about 100 to 200 nucleotides long, while bacterial fragments are often about 1,000 to 2,000 nucleotides long. Replicative DNA polymerases extend the primers by adding complementary deoxyribonucleotides. The leading strand grows continuously. The lagging strand grows one Okazaki fragment at a time. The RNA primers are later removed. The resulting gaps are filled with DNA, and DNA ligase seals the remaining nicks by creating phosphodiester bonds. The notes describe hexanuclease as the enzyme that removes all primers. The exact machinery depends on the organism. In eukaryotic cells, ribonuclease H and flap endonuclease 1 participate in primer removal. Polymerase delta fills the gaps, and DNA ligase 1 seals the fragments. Hexanuclease activity is also central to proofreading, but primer removal and proofreading should not be treated as one identical reaction. Many DNA polymerases proofread while synthesis is occurring. If a wrong nucleotide is inserted, a 3' to 5' hexanuclease activity can remove it so that the correct nucleotide can be added. Additional mismatch repair systems inspect newly synthesized DNA after replication. These layers give DNA replication very high fidelity. At completion, each daughter DNA double helix contains one parental strand and one newly synthesized strand. Replication is therefore called semiconservative. The old strands are not preserved together in one molecule, while an entirely new double helix is produced. Instead, each old strand remains paired with one new complement. When an error escapes proofreading and repair and becomes permanently established in DNA, it is a mutation. A mutation may have no noticeable effect, may alter the amount or function of a protein, may contribute to disease, or may occasionally be beneficial. The notes suggest that diploidy usually protects a child because a second gene copy is present. That is only partly true. A normal allele can sometimes compensate for a recessive loss-of-function mutation, but dominant variants, haploinsufficiency, X-linked disease in males, mitochondrial variants, regulatory mutations, and many other situations are important exceptions. The three major types of RNA. The first major type is ribosomal RNA, or rRNA. Ribosomal RNA combines with proteins to form ribosomes, the structures on which proteins are synthesized. In prokaryotic cells, the major ribosomal RNA species are called 23S, 16S, and 5S. In the eukaryotic cytosol, they are called 28S, 18S, 5.8S, and 5S. The letter S refers to the Svedberg unit, which describes how rapidly a particle sediments during ultracentrifugation. It depends on size, shape, and density, so Svedberg values are not simply additive. Ribosomal RNA makes up roughly 80% of total cellular RNA. It is not merely structural. The catalytic activity of the peptidyltransferase center demonstrates that some RNA molecules can act as enzymes. An RNA molecule with catalytic activity is called a ribozyme. The second major type is transfer RNA or tRNA. Transfer RNA molecules are small, classically described as about 4s, and together account for roughly 15% of cellular RNA. At least one transfer RNA recognizes each of the 20 standard amino acids, and several amino acids have more than one transfer RNA because the genetic code is degenerate. Transfer RNA contains several unusual modified bases and folds through extensive internal base pairing. The familiar cloverleaf drawing shows its secondary structure, while the actual three-dimensional molecule has an L-shaped tertiary structure. Its 3' end carries a specific amino acid through a covalent ester bond. Its anticodon region recognizes a complementary codon on messenger RNA. Transfer RNA, therefore, acts as the adapter that connects a nucleic acid codon to the correct amino acid. The third major type is messenger RNA, or mRNA. Messenger RNA makes up only about 5% of cellular RNA, but it is highly diverse in length and sequence because different messenger RNA molecules encode different proteins. Messenger RNA carries information from DNA to the ribosome. In prokaryotes, one messenger RNA may contain coding information for several proteins. This is called a polycistronic messenger RNA. In eukaryotes, a mature messenger RNA usually encodes one principal protein and is therefore described as monocistronic, although alternative translation products and unusual exceptions exist. Mature eukaryotic messenger RNA also contains untranslated regions at both its 5' and 3' ends. Its 5' end carries a cap made from 7-methylguanosine connected through an unusual 5' to 5' triphosphate linkage. Its 3' end usually carries a poly tail, which is a long sequence of adenine nucleotides. The cap and tail help protect the messenger RNA, support nuclear export, influence translation, and participate in regulation of messenger RNA stability. Other forms of RNA. Precursor messenger RNA is the immature transcript made from a protein-coding gene in the nucleus. It contains exons, which are retained in the mature message, and introns, which are removed during splicing. Splicing is performed by the spliciosome, a large complex containing proteins and small nuclear ribonucleoproteins. Small nuclear RNA is properly abbreviated snRNA, rather than simply sRNA as written in one part of the notes. Small nuclear RNA molecules help recognize splice sites and catalyze the rearrangements that remove introns and join exons. Messenger RNA carries the coding message to the cytoplasm. Transfer RNA delivers activated amino acids to the ribosome. Ribosomal RNA combines with proteins to create the structural and catalytic core of the ribosome. The notes mention about 75 associated proteins. The exact number depends on whether the ribosome is prokaryotic, mitochondrial, or cytosolic eukaryotic, but the central principle is that ribosomes are ribonucleoprotein complexes. Micro-RNA molecules are short single-stranded RNA molecules, commonly about 21 to 23 nucleotides long. They guide silencing complexes to target messenger RNA molecules. Depending on the degree of complementarity, they can reduce translation, promote messenger RNA adenylation and degradation, or occasionally influence other levels of gene regulation. Their best established major role is post-transcriptional regulation, so it is too broad to say that all micro-RNA molecules directly regulate transcription, digestion, and absorption of dietary nucleoproteins. Mononucleotides are essential components of living cells. They are precursors of DNA and RNA, participate in energy transfer, serve as signaling molecules, and form parts of important coenzymes. However, humans do not need intact dietary purine and pyrimidine bases as essential nutrients, because most nucleotides can be synthesized within the body. Dietary nucleic acids are digested in the small intestine. Pancreatic ribonuclease breaks down RNA, while pancreatic deoxyribonuclease breaks down DNA. The resulting oligonucleotides are further processed to mononucleotides. Intestinal phosphatases and nucleotidases remove phosphate groups to form nucleosides. Nucleosidases, or nucleoside phosphorylases, can then split nucleosides into free bases and sugar products. Pyrimidine nucleosides can be cleaved by hydrolytic reactions. Purine nucleoside phosphorylase, or PNP, catalyzes phosphorylasis of several purine nucleosides. This means inorganic phosphate participates in cleavage of the glycosidic bond, producing a purine base and ribose 1-phosphate. Inosine and guanosine are important substrates. Adenosine is handled mainly through adenosine deaminase before subsequent phosphorylasis. The phosphate and sugar components can be reused in metabolism. Some bases can also be salvaged into new nucleotides. Much of the dietary purine load, however, is degraded to uric acid. The intestinal mucosa has active xanthine oxidortictase, which contributes to this conversion. Small amounts of dietary nucleosides and nucleotides may be absorbed and used, but most cellular nucleotide requirements are met through de novo synthesis and salvage rather than direct incorporation of intact dietary nucleotides. Purines from the diet. Diet provides an exogenous source of purines and can influence serum urate. Organ meats such as liver, kidney, and sweetbreads, as well as some fish such as anchovies, have high nucleic acid and purine content. The notes report that strict dietary purine restriction may lower mean serum urate by about 1 mg per deciliter and urinary uric acid excretion by about 1.2 millimoles per day. These are approximate population estimates. The response of an individual patient varies, and urate balance is also strongly affected by kidney handling, alcohol, fructose, medications, body composition, and genetic factors. The notes also state that roughly half of ingested RNA purines and about 1/4 of ingested DN purines may appear in urine as uric acid. The practical lesson is not to memorize those fractions in isolation. The important cause-and-effect relationship is that a higher purine load can increase uric acid production, which can worsen hyperuricemia or gout in a susceptible person. Endogenous sources and the two strategies for nucleotide synthesis. Nucleotide synthesis is medically important because many anti-cancer, immunosuppressive, antimicrobial, and antiviral drugs interfere with nucleotide pathways. A rapidly dividing cell must make large amounts of DNA, so blocking nucleotide production can restrict proliferation. This also explains toxicity in normal tissues with rapid turnover, such as bone marrow and gastrointestinal epithelium. Cells use two broad strategies. In de novo synthesis, the nucleotide is built from small precursors. In salvage, a preformed base or nucleoside is recovered and attached to an activated ribose unit. Purines and pyrimidines follow different construction plans. A purine ring is built piece by piece directly on a ribose scaffold. A pyrimidine ring is assembled first and then attached to ribose. Both strategies initially produce ribonucleotides. Deoxyribonucleotides for DN are then made by reducing the ribose within ribonucleoside diphosphates. This biochemical order supports the evolutionary idea that RNA chemistry preceded DNA chemistry. Thymine is also produced late. The cell first makes deoxyridine monophosphate and then adds a 1-carbon unit to form deoxythymidine monophosphate. Phosphoribacil pyrophosphate, the activated ribose donor. A central intermediate is 5-phosphoribacil 1-pyrophosphate, usually called phosphoribacil pyrophosphate or PRPP. It is made from ribose 5-phosphate and adenosine triphosphate. PRPP synthetase transfers the beta and gamma phosphates of a TP to ribose 5-phosphate. A TP therefore becomes a MP. This consumes the equivalent of 2 high-energy phosphate bonds and creates an activated ribose donor. Ribose 5-phosphate mainly comes from the pentose phosphate pathway. Ribose 1-phosphate released during nucleoside degradation can also be rearranged into ribose 5-phosphate. PRPP is required for de novo purine synthesis, for the attachment step in de novo pyrimidine synthesis, for purine salvage, and for synthesis of several nucleotide-containing coenzymes. The notes contain a typographical error in this section and occasionally write PPP when the intended abbreviation is PRPP. Cellular PRPP concentration is normally kept low through regulation of PRPP synthetase and through rapid consumption by downstream reactions. Magnesium ion, spoken as magnesium 2+, is required as a cofactor. The notes list adenosine diphosphate, 2,3-bisphosphoglycerate, AMP, GMP, and IMP among the inhibitors. Regulatory details vary with the PRPP synthetase isoform, but the overall principle is that abundant purine nucleotides and related metabolites provide negative feedback. De novo purine synthesis, the overall plan. De novo means from the beginning. The liver has a major role in de novo purine synthesis, although the pathway is not limited to the liver. The notes also mention more limited activity in tissues such as the brain and in neutrophils, and the precise capacity varies by cell type. Mature red blood cells lack nuclei and do not synthesize DNA or RNA. They also lack meaningful de novo purine synthesis, so they depend on salvage enzymes, including adenosine kinase and phosphorybosyltransferases, to help maintain their adenine nucleotide pools. The notes identify ribose 5-phosphate and reduced nicotinamide adenine dinucleotide phosphate, or NADPH, as important supporting substrates. Ribose 5-phosphate supplies the scaffold. NADPH is important later for production of deoxyribonucleotides and for regeneration of reduced folate. A phrase in the notes refers to TPP synthesis, but the intended concept is most likely thymidylate, or DTMP, synthesis rather than thiamine pyrophosphate synthesis. The pathway can be divided into 3 large stages. First, ribose 5-phosphate becomes PRPP. Second, PRPP is converted through multiple reactions into inosine monophosphate, or IMP. Third, IMP branches toward either adenosine monophosphate, or a MP, or guanosine monophosphate, or GMP. Building inosine monophosphate step by step. The committed reaction begins when glutamine PRPP amidotransferase replaces the pyrophosphate group of PRPP with nitrogen donated by glutamine. Glutamine becomes glutamate, and the product is 5-phosphorybosilamine, or PRA. This is the rate-controlling committed step of de novo purine synthesis. PRPP activates the enzyme, while IMP, AMP, and GMP provide feedback inhibition. The active metabolite of 6-mercaptopurine also suppresses this pathway. Next, glycine is added to PR by glycinamide ribonucleotide synthetase. This reaction uses a TP and produces glycinamide ribonucleotide, or GAR. Glycine contributes 3 atoms to the future purine ring: carbon 4, carbon 5, and nitrogen 7. The next reaction adds a 1-carbon formyl group from 10-formyl tetrahydrofolate. GAR transformylase converts GAR into formyl glycinamide ribonucleotide, or FGAR. The transferred formyl group eventually becomes carbon 8 of the purine ring. This is one reason folate metabolism is essential for nucleotide synthesis. FGAR then receives another nitrogen from glutamine. FGAR amidotransferase, also called formylglycinamidine ribonucleotide synthetase, uses a TP and converts glutamine to glutamate. The product is formylglycinamidine ribonucleotide, or FGAM. The newly added nitrogen becomes nitrogen 3 of the purine ring. FGAM is then cyclized by a IR synthetase. This ATP-dependent reaction closes the first ring, the 5-membered imidazole ring, and forms 5-aminoimidazole ribonucleotide, or AIR. AIR is carboxylated to form carboxyaminoimidazole ribonucleotide, or CAIR. The notes describe an AIR carboxylase reaction followed by CAIR mutase, with a TP used to activate carbon dioxide. That two-step arrangement occurs in many bacteria. In humans, the bifunctional PAICS enzyme carries out direct AIR carboxylation and the following S-AIC-AR synthetase activity. Human AIR carboxylation is not a biotin-dependent reaction. The carbon introduced here becomes carbon 6 of the purine ring. CAIR then combines with aspartate in an ATP-dependent reaction catalyzed by S-AIC-AR synthetase. The product is succinyl aminoimidazole carboxamide ribonucleotide, or S-AIC-AR. Aspartate contributes nitrogen 1 to the purine ring. Adenylosuccinate lyase removes fumarate from S-AIC-AR and produces aminoimidazole carboxamide ribonucleotide, or AIC-AR. This release of fumarate links purine synthesis with pathways that communicate with the citric acid cycle, also called the Krebs cycle. AIC-AR receives a second formyl group from 10-formyl tetrahydrofolate. AIC-AR transformylase produces formyl AIC-AR, or F-AIC-AR. This formyl group becomes carbon 2 of the purine ring. Finally, IMP cyclohydrolase closes the second ring without a direct ATP requirement and forms inosine monophosphate. The base in IMP is hypoxanthine. The nucleoside inosine consists of hypoxanthine plus ribose, while IMP consists of inosine plus 1-phosphate. This pathway is easier to remember if you understand where the ring atoms come from. Glycine contributes carbon 4, carbon 5, and nitrogen 7. Glutamine contributes nitrogen 3 and nitrogen 9. Aspartate contributes nitrogen 1. Carbon dioxide contributes carbon 6. 10-formyl tetrahydrofolate contributes carbon 2 and carbon 8. In the human pathway, construction from ribose 5-phosphate to IMP uses 5 ATP molecules. 1 ATP becomes a MP during PRPP formation, while 4 become a DP in later reactions. The total cost is therefore 6 high-energy phosphate bond equivalents. The note's total of 6 is best understood as 6 high-energy equivalents, not 6 separate ATP molecules in the human pathway. Branching from IMP to AMP and GMP. IMP is the first complete purine nucleotide in the pathway and the common branch point for adenine and guanine nucleotides. To make a MP, IMP first combines with aspartate. Adenylosuccinate synthetase uses GTP to form adenylosuccinate. This is the regulated step of the AMP branch. Adenylosuccinate lyase then removes fumarate and produces a MP. Aspartate supplies the amino nitrogen that distinguishes adenine. To make GMP, IMP is first oxidized to xanthosine monophosphate, or XMP. IMP dehydrogenase uses oxidized nicotinamide adenine dinucleotide, spoken as NAD+, and water. NAD+ is reduced to NADH. XMP then receives an amino group from glutamine. GMP synthetase uses a TP and water to produce GMP. The two branches show reciprocal energy use. The AMP branch uses GTP, while the GMP branch uses a TP. This cross-use helps balance the two nucleotide pools. A high AMP concentration inhibits adenylosuccinate synthetase, while a high GMP concentration inhibits IMP dehydrogenase. Balanced purine pools are important because DN, a replication, becomes error-prone when one nucleotide is present in gross excess or deficiency. 6-mercaptopurine is converted inside cells into thioinosonate metabolites, including thioinosine monophosphate. These metabolites inhibit glutamine PRPP, amidotransferase, and interfere with the MP and GMP branch pathways. This contributes to the antiproliferative action of thiopurine drugs. Converting purine monophosphates into diphosphates, triphosphates, and deoxynucleotides. A MP and GMP are phosphorylated to their diphosphate forms and then to their triphosphate forms. Adenylate kinase catalyzes the reversible transfer in which a MP and a TP form two molecules of a DP. Guanylate kinase converts GMP to GDP using a TP as a phosphate donor. Nucleoside diphosphate kinase then transfers phosphate groups between diphosphate and triphosphate nucleotides, helping form a TP and GTP. Ribonucleotide reductase acts on ribonucleoside diphosphates, not directly on monophosphates or triphosphates. It reduces the 2' hydroxyl group of a DP, GDP, CDP, and UDP to produce the corresponding deoxyribonucleoside diphosphates. These can then be phosphorylated to the triphosphate forms required for DN, a synthesis. A MP can be converted back to IMP by a MP deaminase. A TP tends to activate this enzyme, while GDP and GTP inhibit it. GMP can be converted to IMP through GMP reductase, with GTP acting as an activator and XMP as an inhibitor. These interconversions help maintain balance between adenine and guanine nucleotides. Regulation of de novo purine synthesis. End products inhibit earlier steps so that purines are not produced needlessly. PRPP availability pushes synthesis forward. Glutamine PRPP, a metatransferase, is the key committed enzyme and is inhibited by purine nucleotides. The pathway then branches, with a MP inhibiting its own branch and GMP inhibiting its own branch. This pattern is called feedback inhibition. The logic is simple. If a cell already has abundant MP, it slows the pathway toward more MP. If it has abundant GMP, it slows the GMP branch. If both are abundant, they work together to suppress the common committed step. This is more useful than memorizing an isolated list of inhibitors. The purine salvage pathway. Salvage recovers performed purine bases, or nucleosides, and converts them back into nucleotides. It is simpler and requires less energy than rebuilding a purine ring from small precursors. Salvaged material can come from normal nucleic acid turnover, diet, or extracellular sources. Purine nucleoside phosphorylase reversibly converts inosine into hypoxanthine plus ribose 1-phosphate, and guanosine into guanine plus ribose 1-phosphate. This reaction is phosphorylasis, not deamination. The notes correctly identify PNP as the bridge between these nucleosides and their free bases, but one degradation table incorrectly labels guanosine to guanine as deamination. Free bases are salvaged by phosphoribosyltransferases, which attach the base to PRPP. Adenine phosphoribosyltransferase, or a PRT, converts adenine into a MP. Hypoxanthine guanine phosphoribosyltransferase, or HGPRT, converts hypoxanthine into IMP and guanine into GMP. Adenosine can be directly phosphorylated to a MP by adenosine kinase. Inosine is not normally phosphorylated directly to IMP. It is first converted by PNP into hypoxanthine, which HGPRT then converts into IMP. Guanosine follows the same two-stage logic. PNP converts it into guanine, and HGPRT converts guanine into GMP. The salvage pathway also suppresses de novo synthesis. It consumes PRPP and produces IMP, AMP, and GMP, which inhibit the committed de novo pathway. A severe HGPRT deficiency therefore does two harmful things at once. It prevents efficient hypoxanthine and guanine salvage, and it leaves more PRPP available to drive de novo purine synthesis. The result is purine overproduction, hyperuricemia, and hyperuricaguria. Complete deficiency causes Lesch-Nyhan syndrome, which also produces severe neurologic and behavioral abnormalities. PRPP synthetase overactivity can also cause excessive purine production and hyperuricemia. These disorders demonstrate why serum urate is linked not only to diet, but also to the rate at which the body synthesizes and salvages purines. Detailed control points in purine metabolism. PRPP synthesis depends on the availability of ribose 5-phosphate and on PRPP synthetase activity. Magnesium 2+ is required. Adenosine diphosphate and guanosine diphosphate are important negative allosteric regulators, and purine nucleotides can provide additional feedback depending on the enzyme isoform. IMP synthesis is controlled mainly at glutamine PRPP amidinotransferase. PRPP activates this enzyme. IMP, AMP, GMP, and their D and triphosphate derivatives signal that purine supply is sufficient and inhibit it. Separate regulatory sites allow adenine and guanine nucleotides to act synergistically. The AMP branch is controlled at adenylosuccinate synthetase, which is inhibited by AMP. The GMP branch is controlled at IMP dehydrogenase, which is inhibited by GMP. The notes also list adenylosuccinate and XMP as feedback inhibitors. In salvage, IMP and GMP can competitively inhibit HGPRT, while AMP can inhibit a PRT. The theme is consistent throughout. Accumulated products slow their own production. Degradation of purines to uric acid. In humans, uric acid is the final major product of purine degradation because humans lack functional uricase, the enzyme that would convert uric acid into allantoin. Adenine nucleotides can reach uric acid through more than one route. In one route, AMP loses phosphate through a 5' nucleotidase and becomes adenosine. Adenosine deaminase removes an amino group and forms inosine. Purine nucleoside phosphorylase removes the ribose component and forms hypoxanthine. Xanthine oxidortictase oxidizes hypoxanthine to xanthine, and then oxidizes xanthine to uric acid. In an alternate route, AMP deaminase converts AMP directly into IMP. A nucleotidase then converts IMP into inosine, after which the pathway proceeds through hypoxanthine-xanthine and uric acid. GMP is degraded by removal of its phosphate to form guanosine. Purine nucleoside phosphorylase converts guanosine into guanine. Guanine deaminase converts guanine into xanthine. Xanthine oxidortictase then forms uric acid. Xanthine oxidortictase can function as xanthine dehydrogenase or xanthine oxidase, depending on its molecular state and electron acceptor. The oxidase form can produce reactive oxygen species such as hydrogen peroxide, spoken as H2O2. Clinically, allopurinol and febuxostat reduce uric acid formation by inhibiting xanthine oxidortictase. This is a useful connection between the biochemical pathway and gout treatment. Transition from purines to pyrimidines. The next page of the notes is headed exogenous source of pyrimidine, but the material immediately under that heading still discusses serum urate, de novo purine synthesis, PRPP, HGPRT, and hyperuricemia. Those are purine concepts, not pyrimidine concepts. The heading appears to be misplaced. The correct takeaway from that repeated material is that increased PRPP availability or reduced HGPRT activity pushes purine production upward and can raise serum urate. We will now move to the true pyrimidine section that follows. The overall plan of de novo pyrimidine synthesis. Pyrimidine synthesis uses glutamine, aspartate, and bicarbonate as major carbon and nitrogen donors. Unlike purine synthesis, the pyrimidine ring is assembled before it is attached to ribose. PRPP therefore enters late in the pathway rather than serving as the structure on which the ring is built. Most reactions occur in the cytosol. The major exception in mammalian cells is dihydrorhodate dehydrogenase, which is associated with the inner mitochondrial membrane. The first complete pyrimidine nucleotide produced is uridine monophosphate, or UMP. The first three enzymatic activities are combined in one large mammalian protein called CAD. The name CAD reflects carbamoyl phosphate synthetase 2, aspartate transcarbamylase, and dihydrorhodase. The last two activities are combined in UMP synthase. which has orotate phosphoribosyltransferase and orotidine monophosphate decarboxylase activities. The formation of carbamoyl aspartate is the first committed step because carbamoyl phosphate can be used in more than one pathway, while carbamoyl aspartate is directed toward pyrimidines. In mammalian cells, however, carbamoyl phosphate synthetase 2 is the major regulated and generally rate-controlling enzyme. The notes call aspartate transcarbamylase both committed and rate-limiting. For human biochemistry, remember committed at aspartate transcarbamylase, but major regulated rate-controlling step at carbamoyl phosphate synthetase 2. The pathway uses 3 ATP molecules to reach UMP if one counts ATP molecules, 2 at carbamoyl phosphate synthetase 2 and 1 to make PRPP. In high-energy phosphate bond equivalence, the cost is greater because PRPP formation converts ATP to a MP. Carbamoyl phosphate synthetase 1 versus carbamoyl phosphate synthetase 2. Carbamoyl phosphate synthetase 1, called CPS1, is located in the mitochondrial matrix and functions in the urea cycle. Its nitrogen donor is free ammonia. It is activated by N-acetylglutamate. Carbamoyl phosphate synthetase 2, called CPS2, is part of the cytosylic CAD protein and functions in pyrimidine synthesis. Its nitrogen donor is glutamine. PRPP promotes its activity, while UTP provides feedback inhibition. Neither CPS1 nor CPS2 requires biotin, even though bicarbonate is used in the reaction. This distinction is highly testable. CPS1 is mitochondrial, uses ammonia, and belongs to the urea cycle. CPS2 is cytosylic, uses glutamine, and belongs to pyrimidine synthesis. Step 1. Formation of carbamoyl phosphate. Cytosylic CPS2 combines glutamine, bicarbonate, and 2 molecules of adenosine triphosphate to produce carbamoyl phosphate. Glutamine supplies nitrogen 3 of the future pyrimidine ring. Bicarbonate supplies carbon 2. The reaction also produces glutamate, 2 molecules of adenosine diphosphate, and inorganic phosphate. This is the most regulated step in mammalian pyrimidine synthesis. PRPP signals a demand for nucleotides and activates the pathway. UTP signals that pyrimidine nucleotide supply is sufficient and inhibits the enzyme through negative feedback. Step 2. Formation of carbamoyl aspartate. Aspartate transcarbamylase transfers the carbamoyl group to aspartate, producing carbamoyl aspartate. Because carbamoyl phosphate is already activated, this condensation does not require another direct input of ATP. Aspartate contributes nitrogen 1 and carbons 4, 5, and 6 of the pyrimidine ring. This is the committed step. Once carbamoyl aspartate has formed, the atoms are directed into pyrimidine synthesis. Do not confuse the word committed with the main regulated step in mammalian cells. They refer to related but distinct ideas. Step 3. Closure of the pyrimidine ring. Dihydrorodase cyclizes carbamoyl aspartate to form dihydrorodate. The notes call this a hydrolytic closure while also describing release of water. The more accurate description is cyclization with elimination of water, a dehydration-type ring-closing reaction. The product now contains the recognizable 6-membered pyrimidine ring. Step 4. Oxidation of dihydrorodate to orotate. Dihydrorodate dehydrogenase oxidizes dihydrorodate to orotate. Orotate is the first fully oxidized pyrimidine base formed in the pathway. In mammalian cells, dihydrorodate dehydrogenase is located on the outer surface of the inner mitochondrial membrane and transfers electrons into the mitochondrial electron transport chain through ubiquinone. The notes show oxidized nicotinamide adenine dinucleotide, or NAD+, being reduced to NADH. That description applies to some soluble microbial enzymes, but not to the human mitochondrial enzyme. For medical examinations focused on human metabolism, remember the mitochondrial, ubiquinone-linked enzyme. Leflunomide is converted to the active metabolite teriflunomide, which inhibits human dihydrorodate dehydrogenase. This reduces de novo pyrimidine synthesis and helps limit proliferation of activated lymphocytes. That is why leflunomide is useful in rheumatoid arthritis. Step 5. Attachment of ribose phosphate. Orotate returns to the cytosolic part of the pathway and receives a ribose phosphate group from PRPP. Orotate phosphoribosyltransferase produces orotidine 5' monophosphate, or OMP, and releases pyrophosphate. Hydrolysis of pyrophosphate by inorganic pyrophosphatase helps make the reaction effectively irreversible. The notes call that enzyme a phosphorylase, but pyrophosphatase is the correct term. OMP is the first pyrimidine nucleotide formed because it contains a base, a ribose sugar, and phosphate. Orotate, by contrast, is only the base. Step 6. Formation of uridine monophosphate. OMP decarboxylase removes carbon dioxide from OMP and forms uridine monophosphate, or UMP. Orotate phosphoribosyltransferase and OMP decarboxylase are the two activities of the bifunctional UMP synthase protein. UMP synthase deficiency causes hereditary orotic aciduria. Orotate accumulates, and the patient cannot efficiently make UMP. The classic presentation includes megaloblastic anemia that does not respond to folate or vitamin B12, failure to thrive, and increased urinary orotic acid without the hyperammonemia expected in a urea cycle defect. Uridine treatment bypasses the block and replenishes pyrimidine nucleotides, producing the RN, a pyrimidine precursors. UMP is phosphorylated by UMP kinase, also called uridylate kinase, to form uridine diphosphate, or UDP. Nucleoside diphosphate kinase then forms uridine triphosphate, or UTP. UTP is the direct precursor of cytidine triphosphate, or CTP. CTP synthetase uses glutamine, ATP, and water. Glutamine supplies the amino group that converts the uracil-type ring into the cytosine-type ring. A common trap is to assume that CDP must be the immediate precursor because CTP contains cytidine. In this pathway, UTP is directly aminated to CTP. UTP and CTP are used in RNA synthesis. Their diphosphate forms also feed the production of deoxyribonucleotides for DNA, producing deoxycytidine triphosphate. CTP can be converted to cytidine diphosphate, or CDP, through normal phosphate transfer reactions. Ribonucleotide reductase then reduces CDP to deoxycytidine diphosphate, or DCDP. A kinase adds another phosphate to form deoxycytidine triphosphate, or DCTP, which is a direct substrate for DNA synthesis. The essential enzyme to remember is ribonucleotide reductase. It converts ribonucleoside diphosphates into deoxyribonucleoside diphosphates. It is tightly allosterically regulated because the cell must maintain balanced supplies of all 4 deoxyribonucleotides, producing the deoxyridine monophosphate intermediate. Deoxycytidine diphosphate can lose phosphate to form deoxycytidine monophosphate, or DCMP. DCMP deaminase removes an amino group and produces deoxyridine monophosphate, or DUMP. DUMP can also be formed from deoxyridine diphosphate. Ribonucleotide reductase converts UDP to DUDP, and phosphatases, or nucleotide processing enzymes, then produce DUMP. The notes call the DCMP deaminase route the major pathway. The relative contribution varies by tissue and metabolic state, so it is safer to remember that both routes feed the common DUMP intermediate, producing deoxythymidine triphosphate. Thymidylate synthase converts DUMP into deoxythymidine monophosphate, or DTMP. The 1-carbon donor is 5,10-methylene tetrahydrofolate. This folate cofactor donates both the 1-carbon unit and reducing power, becoming dihydrofolate in the process. Dihydrofolate reductase, or DHFR, uses NADPH to reduce dihydrofolate back to tetrahydrofolate. Tetrahydrofolate can then be reloaded with a 1-carbon unit to regenerate 5,10-methylene tetrahydrofolate. Serine, through serine hydroxymethyltransferase, is a major source of this 1-carbon unit. Glycine, histidine, tryptophan, and other amino acid pathways can also feed the folate 1-carbon pool, but they should not be imagined as directly attaching a methyl group to DUMP. DTMP is phosphorylated to deoxythymidine diphosphate, or DTDP, and then to deoxythymidine triphosphate, or DTTP. DTTP is the thymine-containing nucleotide used by DNA polymerases. 5-fluorouracil is converted into the active nucleotide fluorodeoxyuridine monophosphate, or FDUMP. FDUMP forms a stable inhibitory complex with thymidylate synthase and the folate cofactor, producing what is sometimes called suicide inhibition. DTMP falls, DNA synthesis is impaired, and rapidly dividing cells are especially affected. The antifungal prodrug 5-fluorocytosine is converted by fungal cytosine deaminase into 5-fluorouracil. Further metabolism produces compounds that disrupt RNA and inhibit thymidylate synthase. Human cells lack cytosine deaminase, which contributes to selective toxicity. Methotrexate and aminopterin inhibit human dihydrofolate reductase, reducing regeneration of tetrahydrofolate and thereby limiting thymidylate and purine synthesis. Trimethoprim also inhibits dihydrofolate reductase, but it has much greater affinity for the bacterial enzyme. Therefore, when the notes list trimethoprim beside methotrexate, remember that the clinically intended target differs. Regulation of pyrimidine synthesis. The notes list regulation at CPS2, aspartate transcarbamylase, OMP decarboxylase, CTP synthetase, ribonucleotide reductase, and DCMP deaminase. For mammalian de novo synthesis, CPS2 is the central regulated step. PRPP and ATP promote activity, while uridine nucleotides, especially UTP, provide feedback inhibition. The source table also lists CTP and UDP as inhibitory signals. The notes table shows aspartate transcarbamylase activated by ATP and GTP and inhibited by CTP. That pattern is classically emphasized in bacterial aspartate transcarbamylase, especially the enzyme from Escherichia coli. In mammalian cells, the corresponding activity is part of CAD, and control is centered mainly at CPS2. This species difference is an important exam trap. OMP decarboxylase can be competitively inhibited by UMP. CTP synthetase requires a TP for the reaction, can be stimulated by GTP, and is inhibited by CTP through feedback. Ribonucleotide reductase is inhibited when deoxyribonucleotide supply is sufficient, with DTTP participating in specificity control and excess D-ATP serving as a powerful overall inhibitor. The notes specifically state that DTTP inhibits ribonucleotide reductase and DCMP deaminase, while DCTP activates DCMP deaminase. These reciprocal effects help balance the cytosine and thymine deoxynucleotide pools. Purine and pyrimidine bases. Purines and pyrimidines are nitrogen-containing heterocycles. A heterocycle is a ring containing carbon atoms and at least one other element, in this case nitrogen. The major purines are adenine and guanine. Other medically important purines include hypoxanthine and xanthine. The major pyrimidines are cytosine, thymine, and uracil. These bases participate in much more than DNA and RNA. Adenine nucleotides are central to energy metabolism. Guanine nucleotides participate in signaling and protein synthesis. Adenosine can act as an extracellular signal and influences coronary blood flow. The same chemical families therefore connect genetics, metabolism, signaling, and physiology. From a base to a nucleoside. Adding a pentose sugar to a nitrogenous base creates a nucleoside. If the sugar is ribose, the product is a ribonucleoside. Adenine becomes adenosine. Guanine becomes guanosine. Cytosine becomes cytidine, uracil becomes uridine. The notes accidentally include thymidine in the list of ribonucleosides of a G, C, and U. That is a mismatch. The RN a pyrimidine U forms uridine. Thymidine is the thymine-containing deoxyribonucleoside commonly found in DNA. A ribose-containing thymine nucleoside can exist and is called ribothymidine, but it is not one of the 4 standard RNA nucleosides listed in basic biochemistry. If the sugar is 2-deoxyribose, the product is a deoxyribonucleoside. The standard DN nucleosides are deoxyadenosine, deoxyguanosine, deoxycytidine, and thymidine, also called deoxythymidine. Atoms in the base and sugar are numbered separately. Pyrimidine ring atoms are numbered 1 through 6. Purine ring atoms are numbered 1 through 9. The sugar carbons are numbered 1' through 5'. The prime mark tells you that the number belongs to the sugar. Thus, the 5' carbon is part of ribose or deoxyribose, not part of the nitrogenous base. The base is attached to the 1' carbon of the sugar through a beta-N-glycosidic bond. In purines, the bond is through nitrogen 9. In pyrimidines, it is through nitrogen 1. From a nucleoside to a nucleotide. A nucleotide is a phosphorylated nucleoside. 1 phosphate produces a nucleoside monophosphate, 2 produce a diphosphate, and 3 produce a triphosphate. The first phosphate is commonly attached to the 5' hydroxyl group of the pentose by an ester bond. Adenosine monophosphate is therefore adenosine plus 1 phosphate. Adenosine diphosphate has 2 phosphates, and adenosine triphosphate has 3. The same naming logic applies to guanosine, cytidine, uridine, and the deoxyribonucleosides. For the standard RNA series, adenine forms adenosine and then adenylate, or a MP. Guanine forms guanosine and then guanylate, or GMP. Cytosine forms cytidine and then cytidylate, or CMP. Uracil forms uridine and then uridylate, or UMP. For DNA, deoxyadenosine forms deoxyadenylate, deoxyguanosine forms deoxyguanylate, deoxycytidine forms deoxycytidylate, and thymidine forms thymidylate. Their monophosphate abbreviations are spoken as DAMP, DGMP, DCMP, and DTMP. The notes suggest that extra phosphates may be added to the same sugar carbon or to another carbon with a hydroxyl group. That wording can be misleading. In ordinary nucleoside diphosphates and triphosphates, such as a DP and a TP, the second and third phosphates form a chain attached to the first phosphate at the 5' position. They are not each independently esterified to different sugar hydroxyl groups. Cyclic nucleotides and bisphosphates are separate structural categories. 3' nucleotides can occur during nucleic acid breakdown, but the common nucleotide triphosphates used in metabolism are 5' nucleotides. In functioning cells, nucleotide D and triphosphates are often more abundant and metabolically active than free bases or nucleosides. Nucleotide functions throughout the body. A TP is the principal immediately usable chemical energy currency of the cell. It is generated through oxidative phosphorylation and substrate-level phosphorylation. A TP supplies energy for muscle contraction, active transport, maintenance of ion gradients, biosynthesis, and many other processes. It also donates phosphate to help generate other nucleoside triphosphates. Nucleotides are the monomeric units of DNA and RNA. Their sequences preserve genetic information, permit replication, guide transcription, and direct protein synthesis. Nucleosides and nucleotides also act as physiologic mediators. Adenosine influences vascular tone and can slow atrioventricular nodal conduction. Adenosine diphosphate promotes platelet activation and aggregation. Cyclic adenosine monophosphate and cyclic guanosine monophosphate act as second messengers. GTP powers G proteins, participates in messenger RNA capping, supports translation, and is required for microtubule assembly. GTP also supports formation of tetrahydrobiopterin. a cofactor needed for aromatic amino acid hydroxylation and nitric oxide synthesis. Several coenzymes contain an adenosine monophosphate component, including oxidized nicotinamide adenine dinucleotide, oxidized nicotinamide adenine dinucleotide phosphate, flavin adenine dinucleotide, and coenzyme A. Nucleotides carry activated intermediates. UDP glucose supports glycogen synthesis and glycosylation. GDP mannose, GDP fucose, UDP galactose, and CMP sialic acid donate sugars during glycoprotein synthesis. The notes say GDP fructose, but the important activated donor in mammalian glycosylation is GDP fucose. CTP helps form CDP choline, CDP ethanolamine, and CDP diacylglycerol for phospholipid metabolism. Nucleotides also serve as allosteric regulators. Their intracellular concentrations tell enzymes whether energy and biosynthetic building blocks are abundant or scarce. This is why a TP, a DP, a MP, GTP, UTP, CTP, and deoxynucleotides repeatedly appear as activators or inhibitors in metabolic pathways. Assembling nucleic acids from mononucleotides. DNA is a polydeoxyribonucleotide, meaning a polymer made from many deoxyribonucleotides. Adjacent nucleotides are connected by 3' to 5' phosphodiester bonds. The 3' hydroxyl group of one sugar is connected through phosphate to the 5' position of the next nucleotide. This creates an unbranched chain with polarity. One end has a free 5' group, and the other has a free 3' hydroxyl group. By convention, a nucleotide sequence is written and spoken from the 5' end toward the 3' end. Polymerases also synthesize new nucleic acid chains in this 5' to 3' direction. RNA uses the same 3' to 5' phosphodiester arrangement, but contains ribose and usually uracil. DNA usually forms a long, base-paired double helix and is chemically more stable because deoxyribose lacks the 2' hydroxyl group. RNA is commonly single-stranded, folds into diverse secondary structures, and is more susceptible to alkaline hydrolysis because its 2' hydroxyl group can attack the phosphodiester backbone. DNA uses adenine, guanine, cytosine, and thymine. RNA uses adenine, guanine, cytosine, and uracil. DNA is the long-term genetic store in human chromosomes and mitochondria. RNA performs many temporary, structural, catalytic, regulatory, and coding roles in the nucleus, nucleolus, cytoplasm, mitochondria, and ribosomes. The source table also compares viruses. The safest principle is that viral genomes may be DNA or RNA, may be single-stranded or double-stranded, and may be linear, circular, or segmented depending on the virus. Human cells, by contrast, use double-stranded DNA as their stable genome and use RNA as intermediates and functional molecules. Chargaff's findings about DNA. In 1950, Erwin Chargaff reported that double-stranded DNA contains nearly equal molar amounts of adenine and thymine and nearly equal amounts of guanine and cytosine. The ratio of adenine to thymine is therefore close to 1, and the ratio of guanine to cytosine is close to 1. However, the ratio of adenine to guanine varies between species. Watson and Crick's double helix model explain these findings. Adenine pairs specifically with thymine, while guanine pairs specifically with cytosine. Every purine on one strand is therefore matched by a pyrimidine on the other. In double-stranded DNA, total purines equal total pyrimidines. Chargaff's equalities do not have to hold in single-stranded nucleic acid. The notes give bacteriophage Phi X174 as an exception because its genome is single-stranded DNA. Without a continuously paired complementary strand, its amount of adenine need not equal thymine, and guanine need not equal cytosine. Final mental summary. Begin with the nucleus. DNA is built from deoxyribonucleotides containing phosphate, deoxyribose, and one of 4 bases. Adenine pairs with thymine, while guanine pairs with cytosine. The order of these bases stores genetic information. During transcription, RNA polymerase uses 1DNA strand as a template and builds complementary RNA in the 5' to 3' direction. Mature messenger RNA leaves the nucleus and reaches a ribosome. During translation, transfer RNA molecules match anticodons to codons and deliver amino acids. Ribosomal RNA catalyzes peptide bond formation, and the ribosome builds a protein until it reaches a stop codon. Before a cell divides, both DNA strands are copied. Helicase opens the helix, primase supplies RNA primers, and DNA polymerases extend new strands only in the 5' to 3' direction. This produces a continuous leading strand and a discontinuous lagging strand made from Okazaki fragments. Primer removal, gap filling, ligation, proofreading, and repair create two semi-conservative daughter molecules, each containing one old strand and one new strand. The cell must continually supply nucleotides for all of this work. Purines are built directly on PRPP until IMP is formed, and IMP then branches to a MP or GMP. Purine bases can also be salvaged through a PRT and HGPRT. Purines that are not reused are degraded through hypoxanthine and xanthine to uric acid. Pyrimidines follow the opposite construction logic. Their ring is built first from glutamine, bicarbonate, and aspartate. The pathway forms orotate, attaches it to PRPP to make OMP, and decarboxylates OMP to UMP. UMP gives rise to UTP and CTP for RNA. Ribonucleotide reductase produces deoxyribonucleotide precursors for DNA, while thymidylate synthase and folate chemistry convert DUMP into DTMP. The language at the end is simple but essential. A base is only the nitrogenous ring. A nucleoside is a base plus sugar. A nucleotide is a nucleoside plus one or more phosphates. Nucleotides join through 3' to 5' phosphodiester bonds to form nucleic acids, and their many additional roles connect genetics with energy metabolism, signaling, coagulation, membrane synthesis, and enzyme regulation. High-yield memory pearls. DNA uses deoxyribose and thymine, while RNA uses ribose and uracil. Adenine pairs with thymine in DNA, and guanine pairs with cytosine. The common B form of DNA has approximately 10.5 base pairs per turn. Genetic information is read in 3 base codons, and the template strand must not be confused with the coding strand. Transcription makes RNA from DNA. Translation makes protein from messenger RNA. RNA polymerase uses energy-rich ribonucleoside triphosphates, and the ribosome's peptidyltransferase center is mainly catalytic ribosomal RNA. A start codon is usually a UG, while the stop codons are UAA, UAG, and UGA. DNA polymerases synthesize only from 5' to 3'. The leading strand is continuous, while the lagging strand contains Okazaki fragments. DNA, a replication, is semi-conservative. In eukaryotic cells, Okazaki fragments are about 100 to 200 nucleotides long, not merely several bases long. Ribosomal RNA makes up roughly 80% of cellular RNA, transfer RNA a roughly 15%, and messenger RNA a roughly 5%. Svedberg values describe sedimentation and are not arithmetically additive. Precursor messenger RNA contains introns and exons, and small nuclear RNA helps perform splicing. PRPP is the activated ribose donor shared by de novo and salvage pathways. De novo purine synthesis builds the ring directly on PRPP and first forms IMP. The committed enzyme is glutamine PRPP amidotransferase. IMP becomes a MP using GTP and aspartate, while IMP becomes GMP using NAD+, ATP, and glutamine. HGPRT salvages hypoxanthine into IMP and guanine into GMP. A PRT salvages adenine into a MP. Severe HGPRT deficiency causes Lesch-Nyhan syndrome and increases uric acid production. Human purine degradation ends in uric acid, and xanthine oxidortictase is inhibited by allopurinol and febuxostat. Pyrimidine synthesis builds the ring before attaching it to PRPP. CPS1 is mitochondrial, uses ammonia and functions in the urea cycle. CPS2 is cytosolic, uses glutamine and functions in pyrimidine synthesis. Human dihydrorhodate dehydrogenase is mitochondrial and ubiquinon-linked. UMP synthase deficiency causes orotic aciduria that can be treated with uridine. Ribonucleotide reductase acts on nucleoside diphosphates. Thymidylate synthase converts DUMP into DTMP using 5,10-methylene tetrahydrofolate. 5-fluorouracil inhibits thymidylate synthase through FDUMP. Methotrexate inhibits human dihydrofolate reductase, while trimethoprim preferentially inhibits bacterial dihydrofolate reductase. Finally, remember the naming ladder. A base plus a sugar is a nucleoside. A nucleoside plus phosphate is a nucleotide. Nucleotides join through phosphodiester bonds to form DNA or RNA. In double-stranded DNA, adenine equals thymine and guanine equals cytosine, but Chargaff's equality does not necessarily apply to a single-stranded genome.